Okay, my voice clear? Audio video clear or not? Send your comments. Okay, okay. Yes. Now okay. Okay, friends. So earlier I given my introduction. Again, listen once again. So actually, till now, mathematical part we are discussing in physics, because to understand the physics, mathematics is required. That's why medical students, those who prepare for NEET, they struggle in physics. They get less marks in physics. Why? Because there is the role of maths to understand the physics. Okay, that's why first we started mathematical topic only, so that you can understand physics properly. And these are helpful for you complete 11th and 12th in both year, right? Okay, so I will teach you this trigonometric functions, and my name is Tulsi Ram Darwal and I done my BTech from IIT Roorkee. Have you know about IIT? You know, IITs are famous in all over world, not our in India, right? Okay, and you can call me TRD sir, right? Short is TRD sir. Okay, so three things I will discuss. First, these things you have to know before starting your preparation. What these are? First point, you are preparing for NEET. So actually, you given your 10th exam. Okay. Now, some of your friends, they are taking this mathematics, and you are taking this biology or preparing for NEET, or your friend they selected maths, they will prepare for IIT. Okay, till now your friend and you, both you studied equally, right, till 10th. Now one is choosing for IIT and one is for NEET. So what is the difference between IIT and NEET? First we have to know this thing. Why? Because nowadays you can see competition in India. Day by day it is increasing. This year 25 lakh students registered for NEET exam, and almost 15 lakh student registered for IIT JEE main. Okay, so in this competition, how we will be selected? For that you have to put your each step by knowing everything clearly. Okay, so what to know? First point about the exam. You will write NEET exam, your friend will write IIT. Okay, what is the difference between them? So very big difference is what? Listen, all of you careful. Listen. IIT JEE cutoff marks is only around 35%, 33%, 36%. How much? 35%, 36%, like that. Okay. And NEET cutoff marks last year in AP, Andhra Pradesh, out of 720, those who got 550 plus marks, they got government college. Means it is around how much percent? Around 80%, right? So here around 80% cutoff in NEET, and in IIT JEE cutoff marks only 35%, 33%. Now you can say, "Arey sir, this NEET is then very tough." No, actually NEET paper, what NEET paper demand from you, that we have to know. So NEET paper demand speed and accuracy. Speed and accuracy. Okay. Why? Because in NEET, total 180 questions you have to solve in 3 hours. Okay. And in JEE Advanced, IIT JEE, total around 60, sometime 56, sometime less also, 54 also. So in IIT JEE, around 60 question you have to solve in 3 hours. Okay, so 3 hours, 60 question, 1 question 3 minutes. In NEET, 180 question 3 hours, means 1 question 1 minute. This is the difference. Okay, so speed is required for NEET. For IIT JEE, speed is required, but not so much. What is required there? Depth study of concepts. Because why cutoff marks is very low in IIT JEE? Because their paper level is very tough. Very logical questions they ask. Depth concepts they ask. So depth study is required for IIT JEE, but for NEET that much depth study is not required. Questions come simple. Out of, if we talk about physics, so out of 45 questions in physics, total 45 questions. So out of 45 questions, only 5 or 4 5 we can say questions are tough. Otherwise remaining questions are simple, right? So this is the difference. There they ask in NEET simple question. That's why speed and accuracy. Accuracy means sometimes we do silly mistakes. In calculation we do mistake. 2 into 3, 6, we know it. But in exam we write 5. Why? Because in our mind what is running? Addition is running. 2 plus 3, 5. So like that mistakes we do. So these mistakes are not acceptable in NEET exam because cutoff marks is very high. If you lose 2-3 questions, these silly mistakes you did there means for you trouble is there, because maybe you will not get that much marks, right or not? So students, very important point is what? Very important point is what? That we have to accurate and speed, both are necessary for NEET. Understood? Okay. Now, how to improve the accuracy? How to improve the speed? The speed is improved by practice. If you will do as much practice, then your speed will increase. Okay. Accuracy, when we are understanding anything, at that time we have to listen each and every point very carefully, then only your chance of mistakes will be very less, right? Okay, so these 2 points you have to remember. Understood? Now this, I already explained you that this mathematical part will help you to understand the physics. If you are weak in mathematics, then you will struggle in these 2 years. And if you will struggle in physics, means you will not select, because physics only matter. Everyone can understand chemistry. Most of the students get good marks in chemistry and in biology. Where they struggle? Only in physics. So basic is what? Mathematical part. Mathematical part if you understood, then you will start to understand physics also eventually. Understood? So till now, am I clear? Whatever I said, you understood? Send comments. Yes sir, we understood, like that. Okay students. Now we will start it. Okay, yes, some students, they are sending their response. Very good, very good, very good. Okay, so students, now like our session and come back to our topic, trigonometric functions. This topic is familiar with you. You already know about it. You know about sine theta, cos theta, tan theta, cot theta, sec theta, cosec theta. You know it? Yes. But what too special here? understand. Special is what? Actually, these trigonometric functions we defined for right angle triangle. We defined these trigonometric function for right angle triangle. Okay, this is right angle triangle. Now in right angle triangle, you know, this is base and this is what? Perpendicular. Okay, and this is what? Hypotenuse. Okay, now this angle is theta. This angle is theta. You know, unit of theta we measure angle in degree. Okay, one more unit is there, we will discuss about that also. So this angle is theta. We call this side adjacent side also, and this side is opposite to this angle. This side is opposite to this angle, so this side is opposite side. Okay, this is adjacent side and this is opposite side. Now you know that this perpendicular square and base square equal to hypotenuse square, that also you know. So p square plus b square equal to h square. Perpendicular square plus base square equal to hypotenuse square, you know it. Okay. Now here we define trigonometric functions. How we defined? Understand. Sin theta, first trigonometric function sin theta. This sin theta is equal to perpendicular divided by hypotenuse, p by h, that also you know. Then cos theta is base by hypotenuse. Okay. Then tan theta, these are important. We talk about these functions frequently. Tan theta is perpendicular by base. Okay, these are basic functions. Other are also there. Sin theta is equal to 1 by cosec theta. Cos theta is equal to 1 by sec theta, and tan theta equal to 1 by cot theta. Okay, now tell me, till now you know everything? Till now you know everything? Okay. We understood. Now next is, yes. Okay students, so you know these things. Now here we know, if we do sine square, if we do here sine square and cos square, and if we add them, so what we will get? Sine square theta and cos square theta, what we will get? So here p square by h square, then cos theta b square by h square. One more point here. Actually in physics, what you have to do, focus more on derivation. Do not remember everything, do not try to remember everything, try to remember the concepts, try to remember the derivations, how it is coming, that you have to focus more, then your physics will become strong. Okay, so sin theta perpendicular by hypotenuse, cos theta base by hypotenuse, we are adding them. Okay, so here LCM is h square, so it will be p square plus b square. Okay, now you know p square plus b square how much? H square. So this is h square, so h square by h square is 1, right? So very important point, that sin square theta and cos square theta, their sum is equal to 1. So this is the relation that you have to remember, because directly we use sin square theta plus cos square theta equal to 1. Then next relation is 1 plus 10 square theta. This 1 plus 10 square theta is equal to sec square theta. How? We can derive like that only. Put tan theta value p by b. So 1 plus tan theta value p by b. So it is p square by b square. Then take LCM. It become b square plus p square by b square. Okay. And we know p square plus b square is what? H square. So h square by b square. So cos theta is b by h. So sec theta is h by b, right? So here this will become sec square theta. Okay. Understood? Understood? Okay. So first this relation. Then we understood this relation. One more relation, that is 1 plus cot square theta. This is equal to cosec square theta. 1 plus cot square theta is equal to cosec square theta. Now cot theta we can derive here. Cot theta is, if tan theta p by b, which is 1 by cot theta, so cot theta will be b by p. So 1 plus b by p. Take LCM, then it become p square plus b square by p square. This is what? This is what? Hypotenuse square, h square by p square. So you know p by h sin theta. Cosec theta is h by p. Cosec theta is h by p. So h by p cosec theta square is there, so it become cosec square theta. Clear? So these three relation we understood. Sin square theta plus cos square theta 1. 1 plus tan square theta sec square theta. 1 plus cot square theta cosec square theta. Okay. Now next is angle value. So I think you know sin 0 value. What is the value of sin 0? Can you tell? Sin 0 degree value is how much? Sin 30 degree value is how much? Send your comments. I will check. Yes, very good. Many students are sending 0. Yes, correct. Sin 0 is 0. Sin 30 is how much? Sin 30 is how much? 1 by 2. Very good. Very good. 1 by 2. Some students are sending 1. That is wrong. 1 is wrong. 1 by 2 is correct. Okay. Now send your comments. Sin 45 is how much? Who will send first? Sin 45. This girl Jinu. She sent first. Okay. Okay. Many Venkat Reddy. Okay. Bhanu. Okay. Santosh. Very good. Very good. Sin 45 1 by root 2. Okay. Then sin 60 degree. Sin 60, I think you know these values, root 3 by 2. And sin 90 is 1. These angle you know, right? Now, these angles you know. Now, if suppose anyone asks you that find value of sin 15 degree, find value of sin 15 degree. Sin 15 degree. So can we calculate sin 15 degree value? Do you know sin 15 degree value? So till now all you know, but now my question is, can you calculate sin 15 degree value? Okay, one student is sending Samrud and this Savneet. Very good. These okay, some students are saying, sir, we don't know, and some are saying that yes, we can do how? That 15 degree is 45 degree minus 30 degree. But actually it is, how to do it? Directly we can't do. Don't think that 15 degrees 45 minus 30. 45 value is sine 45 value 1 by root 2 and sine 30 value 1 by 2. So it will be 1 by root 2 minus 1 by 2. It is wrong. It is not correct. So how to do? So actually a formula is there. We have to use that formula. What is that? A plus B, sine A plus B. We can break a angle into two angles that we know. Sine 15 degree, or suppose if anyone asks that find sine 75 degree value. Okay. So we can break this 75 into two angle. Those, which angle? Those angles, their value we know. 75 we can break 45 plus 30. 45 also we know and 30 also we know. So we can break this 75 like this, sine 45 plus 30. Okay, sine 45 plus 30. Because we know 45 degree value and 30 degree. So we can break a angle into two angle which we know. Okay. So we can use there this formula sine A plus B. Now this formula is okay. Now this formula is sine A plus B formula. This formula you have to remember. I think it is new for you. Sine A cos B plus cos A sine B plus cos A sine B. This formula you have to remember, sine A plus B formula. Sine A plus B equal to sine A cos B plus cos A sine B. This formula you have to remember. We can break a angle into two non-angle. 75 we can break 45 plus 30. Then sine A plus B formula is sine A cos B plus cos A sine B. First sine A, then cos A. Here cos B and then sine B. Understood? And if suppose minus is there, suppose this formula is A minus B. Minus is there, then everything will be same. Only at the place of plus sign, minus will come. So it will be sine A cos B minus sine B cos A. Okay. So sine A cos B minus sine B cos A. Understood? Sine A minus B formula is sine A cos B minus cos A sine B. So what is the difference? Only if here plus, so here plus, here minus, then minus. Understood? Okay. So this 75 we can break 45 plus 30. Then A is now 45 and B is now 30. Understood? Okay. And one more point. 15 degree. How to find 15? So we can break this 15 degree like this. 15 degree, how we will break? those angle which you know, 45 minus 30. 45 minus 30. Because 15 we will get by difference, 75 we will get by addition. Understood? Okay. So here now we can use this formula. So can we calculate now value of sine 75 and 15 degree? Yes. So first this sine 75 degree. So it is sine 45 plus 30, right? So A is 45, B is 30. Use the formula. The formula is sine A is 45, cos B is 30, then cos A, to cos A is 45, okay, and sine B is 30, to sine 30. This is in multiply, okay. Here this is in multiply 30. Now put the values. Sine 45 value is how much? 1 by root 2. Cos 30 value root 3 by 2. Okay. And cos 45 1 by root 2. Then sine 30, it is 30. Here it is 30, okay. This is sine 30. So sine 30 value is 1 by 2. Okay. Now take LCM. So this is root 3 by 2 root 2. And this is 1 by 2 root 2. Take LCM. LCM is how much? 2 root 2. And here root 3 plus 1. Root 3 plus 1. Okay. So actually some students are saying that it is 0.15 like that. But value is root 3 plus 1 by 2 root 2. This is our answer. Understood? So how to find unknown angle value? And we cannot find any angle value. Suppose if you ask, sir, find the value of sine 43. So sine 43 we don't know. We can only find those angles which we can write in terms of known angle. Right? 75. 75 we can write 45 plus 30. 15 degree we can write 45 minus 30. These angle we can calculate, but sine 43 we can't calculate. Understood? Okay. So we understood here. Now sine 15 degree, this is your very good. Some students are very sincere. They already calculated sine 15 degree, root 3 minus 1 by 2 root 2. Very good. Very good. Some students are very sincere students. Very good. Sine 15 degree value is, you have to calculate it. This is homework for you. Okay. I already explained you that we have to break 15 degree. How? 45 minus 30. Then use this formula. A is 45 and B is 30. Put the values, you will get answer. Okay. So sine 15 degree value is root 3 minus 1 by 2 root 2. Here plus and here minus. Understood? Okay. So we understood that how to find those angle values which we don't know, like 15 degree and 75 degree, like that. Okay. Now next is this formula we understood for sine function only. This formula is for cos function and tan function also there. So that also we have to know. Okay, so this part completed. Now next is we will discuss this formula for cos theta and tan theta. So this part completed. Now I am moving on next slide. Okay, so we understood sine A plus B formula, that is sine A cos B plus cos A sine B. Why again I am writing it? Because here we have to understand this point also. Suppose if A and B both are equal. Suppose A and B both are equal. 90 degree. We can break 90 degree into 45 because 45 value we know, 45 value also we know. Now you will say, sir, why to break? Already we know sine 90 is 1. Actually, suppose 120 degree. Do you know sine 120 degree value? Do you know sine 120 degree value? Send your response. Yes. So we don't know, but we can break it. How? 90 plus 30, otherwise 60 plus 60, right? So here we can break this angle into two equal angle also. So if A and B both are equal, and suppose they are equal to theta, so it become here sine theta plus theta. So this is how much? Sine 2 theta, right? So put here theta. A is theta, B also theta. So it becomes sine theta cos theta. Again cos theta sine theta. So both are equal, na? Sine theta cos theta, sine theta cos theta. So it becomes 2 sine theta cos theta. So this formula also you have to remember. By A plus B only we got this formula. Sine 2 theta equal to 2 sine theta cos theta. Is it clear? Okay, sine 2 theta equal to 2 sine theta cos theta. Understood? Now, next is similar like I explained you that cos theta formula also we have to know. So for cos function, A plus B formula is cos A cos B. This formula you have to remember. And actually here one twist is there. What? In sine theta formula, if plus is there to here plus, here minus is there to here minus. But in cos theta formula it is different. Here plus is there, but here minus. So this twist is there, you have to remember it. Okay. In cos A plus B, cos A cos B minus sin A sin B. We understood it. Now if suppose here minus is there, then to plus, then minus, to minus, then plus. So it will be cos A cos B plus sin A sin B. Okay, we understood it. So cos theta formula also we understood. Now next is here also 2 theta formula. Sin 2 theta we understood. Now if anyone asks, find cos 2 theta formula. So how to find? So here if A and B both are equal, cos A plus B na. If A and B both are equal, A is equal to theta, B also equal to theta, then it become cos theta plus theta, right? Means cos 2 theta. Agree or not? Put here A value theta, B value theta. So cos theta into cos theta, cos square theta. Sin theta into sin theta, sin square theta. So cos 2 theta formula is cos 2 theta formula is cos square theta minus sin square theta. So this formula you have to remember. Is it clear? Now here we can change this formula into other form also. How? Already we discussed what? That sin square theta plus cos square theta is 1. Yes. So here we can convert this formula completely into sin theta or completely into cos theta, right? Because one thing we know that sin square theta plus cos square theta equal to 1. So at the place of cos square theta, what we can put? At the place of cos square theta, we can put 1 minus sin square theta. Otherwise, at the place of sin square theta, suppose we have to find out completely in terms of cos theta. So at the place of sin square theta, we can put here at the place of sin square theta value 1 minus cos square theta. Right or not? So we can completely convert this formula into cos theta, otherwise into sin theta. If we want to convert into cos theta, then sin square theta we have to eliminate. Sin square theta value 1 minus cos square theta put here. So it become cos square theta minus sin square theta value 1 minus cos square theta. So this minus minus become plus. So cos square theta plus cos square theta, 2 cos square theta. 2 cos square theta minus 1. Understood? Okay. Here also we can convert completely in terms of sin square theta. How? Sin square theta value is, if we want to convert in sin square theta, so we have to eliminate cos theta. Cos square theta value 1 minus sin square theta. Put here. So cos square theta value is 1 minus sin square theta, and this minus also. So it become how much? Minus sin square theta minus sin square theta minus 2 sin square theta. Right? So cos 2 theta is equal to 1 minus 2 sin square theta. Is it clear? So only for cos theta, specifically only for cos theta, we discuss 3 formula. Now you will say where to use these formulas? We will use in physics only. Cos 2 theta formula 1 minus 2 sin square theta. Otherwise, completely in terms of cos theta formula is 2 cos square theta minus 1. And in terms of both cos theta and sin theta, this is the formula, cos square theta minus sin square theta. Understood? So for sin, we only understood only in one form, sin 2 theta equal to 2 sin theta cos theta. But for cos 2 theta, 3 formulas, cos and sin both, only cos and only sin. Understood? Is it clear to all of you? Okay. So we understood sin A plus B formula, sin A minus B formula. Then we discussed cos A plus B formula, cos A minus B formula, and finally now tan theta formula. Okay. So tan A plus B formula is, so first point, all of you know tan theta is sin theta by cos theta. Right? So we can write this tan A plus B equal to sin A plus B by cos A plus B. Both formula you know. By using them you can calculate. One point. Another point, directly formula, that result, final result I am writing here. Final result is tan A plus tan B by 1 minus tan A tan B. This is tan A plus B formula. Here plus plus, but in divide minus is there. And now you can ask, sir, why here in sin theta and cos theta Nothing is in divide, only in tan theta divide is there. Why? Because by that only it came. Tan theta sin theta by cos theta. That's why in divide this quantity is there. Understood? Okay. Now tan A minus B formula. So here plus plus, in divide minus. So in minus, if tan A minus B, then here it will be tan A minus tan B by 1 plus tan A tan B. Here minus to minus, but in divide plus, reverse. Understood? Okay. Now here also 2 theta formula we have to know. So how we will get 2 theta if A and B both are equal? A and B both are equal. So theta plus theta, 2 theta. So it will be tan theta plus theta. Then it become tan 2 theta. Can you write tan 2 theta formula? All of you. Write final formula of tan 2 theta. Send your comments. Write final formula of tan 2 theta. Send your response. Okay, I am waiting. Send your response. Okay, very good, very good. Some students, they send their answer. Very good, correct. So if theta and theta we will put here, so it will be tan theta plus tan theta. So how much? 2 tan theta. Yes. Okay. Then here also theta. So tan theta, it become tan square theta. To divide by 1 minus tan square theta. Understood? So this is the formula of tan 2 theta. So we discuss all the formulas: sin A plus B, sin A minus B, cos A plus B, cos A minus B, tan A plus B, tan A minus B, sin 2 theta, cos 2 theta, tan 2 theta. Okay. Now final question. Calculate value of tan 75 degree. Calculate value of tan 75 degree. Send your answer. Let's see who will give first. Send your answer. Find tan 75 degree value. That I know. Some students are sending 45 plus 30. Arre, everyone know it. Final value I am asking. One student sent 2 plus root 3. Okay. Arre, tan 45 plus 30 everyone know. Final value, send your final answer. One student sent 2 plus 3. Arre, not 2 plus 3 is wrong. Root 3 plus 1 by root 3 minus 1. One student sent this. Okay. 2 plus root 3. 2 plus root 3. Yes. 2 plus root 3. Most of you are sending 2 plus root 3. So yes, this I think this is correct. We can find here. We can write 75, 45 plus 30. Now use the formula. Formula is tan A plus B. This is A, this is B. So tan 45 plus tan 30. Then 1 minus tan 45 tan 30. Tan 45 value 1. Tan 30 value 1 by root 3. Here 1 minus tan 45 1. This is 1 and tan 30 1 by root 3. Okay. Now take LCM. It will be root 3 plus 1. Here also take LCM. Root 3 minus 1. So it become root 3 plus 1 by root 3 minus 1. Now some students, they are intelligent. What they did, here we can multiply this value by root 3 plus 1 and divide root 3 minus 1. Okay, rationalization. So it become how much? Sorry, here plus. So it become how much? You know, a minus b into a plus b is a square minus b square. So it become a square minus b square. A is root 3 squared 3 minus b square, b is 1, 1 square 1. So it become 3 minus 1, and here root 3 plus 1 whole square. Root 3 plus 1 whole square. We can open this square. If we open it, to what it will become? Root 3 square 3, then 1, then 2 root 3. Divide by 3 minus 1 2. So it become 3 plus 1 4, 4 plus 2 root 3 by 2. So divide 4 by 2 2, 2 root 3 by 2 root 3. So final answer 2 plus root 3. So some students, they send their answer, and this answer also I seen. Yes, 2 plus root 3 also there. So correct answer is 2 plus root 3, but this is also correct. Root 3 plus 1 by root 3 minus 1. Okay, so yes, this class, I can say now that it is fruitful because many of you given correct answer. Okay, so we learn that if angle is not given directly like 30 and 60, 45, 75, 15, such that angles are given, so those value also we can calculate. We learned these formulas. Okay friends, so thank you for listening me. Now chemistry class will be there.
Yes, good morning everyone. Good morning beta. Is it audible and visible to you? Is it audible and visible to you? Good morning, good morning. I hope you're doing well. Right. So in previous class we have done our topic concentration terms, right? So today we are starting with organic chemistry, or I will say basic organic chemistry, right? Very simple, and you know most of it because it is connected to your 10th class. Okay bacha? Okay, very good. So we are starting our basic organic chemistry. Okay bache? Fine. Everything is audible. Okay. Okay, fine. First of all, let us understand what is organic chemistry. Right bache? You know, if you compare all the elements in your periodic table, so carbon make lot of compounds. Carbon is forming million, trillion, many compounds on this earth, right? So you see, carbon is some special element which is making so many compounds. So there are so many compounds, so that we have to keep compounds of carbon in a separate category, right? We call this organic chemistry. Okay, so what is organic chemistry? I will say once again. Organic chemistry is study of carbon compounds mainly, right? And connected to mainly living organisms, or you can say plants like this. So study of carbon compounds mainly is our what? Organic chemistry. Right, bache? Clear? So we will see, if I give a simple definition, right? So I can say, what is organic chemistry? Already I have written definition. But see, I'm adding one more. So you can say study of compounds, or you can say study of hydrocarbons, basically you can say, right? Study of hydrocarbons. Study of hydrocarbons and their derivatives. Study of hydrocarbons and their derivatives. Fine. Now question come to our mind. Sir, what is hydrocarbon, right? Tell me, bache, if any one of you can tell me, what is the meaning of hydrocarbons mainly? What is the meaning of hydrocarbons? Means mainly compounds of carbon and hydrogen, right? So I will say compound of carbon and hydrogen. You can say compound of carbon and hydrogen, right? After carbon, if I say, after carbon, hydrogen will be the, like, will again repeat in our hydrocarbons or in our organic chemistry. Okay, compounds of carbon, and after carbon, what will come? Hydrogen again, right? So study of what? Hydrocarbons is organic chemistry. Clear, bache? Right? And as I said, carbon and hydrogen are the definitely most important, right? After this, as I said, derivatives, right? Derivative means some other elements can also come sometime, right? Some other elements can come, like I can say, sometime you will find oxygen, sometime you will find what? Nitrogen, or sometime you will find what? Sulfur, etc. So derivative of hydrocarbons will contain oxygen or sulfur or nitrogen or some other elements. Okay, maybe phosphorus type of things, right? I'm saying once again, organic chemistry is the study of hydrocarbons, or you can say mainly compounds of carbon, and mainly these are connected to our living worlds, or like plants, or you can say, right? Clear, bacha? Very good. Here also we have written, so it is what? Chemistry in which we are studying what? Structure, properties, composition, reactions, and preparation of what? Organic compound. Clear, bacha? Clear? Organic compound, example if I say, it is what? Glucose, sugar, alcohols, thiols, carboxylic acid, or you can say your amines, haloalkanes, right? So many things are there. So we will study step by step. Clear, bache? Okay. So first of all, the next thing is, why, give me the reason why only carbon is having so many compounds. Why only carbon is making so many compounds, right? The reason is its high catenation power, right, bache? Yes, many students are giving right answer. So why only carbon is making so many compounds, right? So because of its high catenation power. High catenation power of carbon, right, enables it to form so many compounds. Okay, bacha? So if someone asks you why only carbon is making so many compounds, why not other elements? So I will say the reason is catenation power, right? Many are writing the right reason, the catenation power, right? Now, if you are not aware what is the catenation power, right? So I am writing here, what is catenation power, beta? What is the meaning of catenation power? So you can say in simple words, self-linking tendency, means to connect with same type of elements. I will say self-linking, you can say tendency. You see, carbon is very much interested in making bond with other carbon. So you will see carbon bond is very strong bond. It's a strong bond, right? That's why carbon is very much interested in making bond with other carbons, and that's why carbon have very high catenation tendency, and that's why because of high catenation it is making so many compounds. Very good, very good. So if I say here one carbon is connected to beta how many carbons here? See, 4 carbons. Or if I say here, look at this beta, I am writing here CH3, then what is it? CH2, 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 CH3. So carbon is making a very strong bond with the other carbon. That's why it has very high tendency of self-linking. Means carbon with carbon, right? And because of that reason, it have high catenation tendency, and that's why it is making so many compounds. I hope we are clear. I hope we are clear. Right, bache? Right? Clear? Okay. Very good. Very good. Now, just tell me, you have done your 9th, 10th class and this topic is connected to that part only, right? Just tell me, carbon normally in its compounds make how many bonds? Carbon normally in its compounds make how many bonds? Options are 1 bond, 2 bond, 3 bond, 4 bond. Carbon normally make how many bonds in its compounds? Very good, very good, very right. 4, 4, 4, 4, very good. Clear, bacha? You will see a small point I will say, tetravalency. Right? Tetravalency of carbon. Clear, bacha? So carbon will make 4 bonds in most of its compounds. Will make 4 bonds in most of its compounds, right? Clear, bache? Right? So you see here, you see carefully, like if I am saying here, I am making a compound, like this is a compound. If I am showing, I will use this in an entire lecture, so please be careful. If I am showing this carbon's only 1 bond, if I am showing this carbon's only 1 bond, so that means it is connected to what here? 2 hydrogens, or sorry, 3 hydrogens, right? Overall 4 bonds will be present. If I am showing 1 bond with carbon, the other 3 bonds are what? With hydrogen. We will not show hydrogen again in every compound. So it is understood that if I am showing only 1 bond, the remaining bond are what? Hydrogen. Very good, right? This carbon I am showing how many bond? 2 bond. So remaining 2 are what here? Hydrogen, right? I am showing its 2 bonds, so remaining beta how much? 2 hydrogen, right? Very good. And here how many? I am showing 1 bond, so remaining 3 bonds it will make with what? Hydrogen. Clear? So you can see clearly, carbon is making 4 bonds. Every carbon here is making 4 bonds. In complete organic chemistry, most of the times, in 99.9%, carbon will make 4 bonds, right? 1 or 2 exceptions possible in carbocations, carbanions, but normally, right? Clear, bache? Now, just tell me, if I am showing this is carbon, this is carbon, this is carbon, this is carbon, like this, I am making some cyclic structure, right? Just tell me, this carbon I am showing here 2 bonds, right? For this carbon I am showing 2 bonds, right? So how many hydrogen are present here? This carbon is having 2 bonds with other carbons. So how many hydrogen are present on this carbon? Is everyone fast? Very good, very good. This carbon is making 2 bonds. So definitely to make total 4 bonds, it will have here 1 hydrogen and other hydrogen, right? So it is understood. Very good. It is also making what? 2 bonds with hydrogens. Here also 2 hydrogen are present. Here also hydrogen present. Here also 2 hydrogen, and here also what? 2 hydrogens. Very good. Clear? So I am saying once again, tetravalency of carbon means carbon will make 4 bonds in its compounds to complete its octet, right? Definitely you remember about octet, okay? Carbon will make total 4 bonds. Are we clear about this? Okay, very good. Next thing I am moving to the classification. We'll come step by step to everything, right? Next bacha is our classification of organic compounds. Next topic is classification of organic compounds. Fine? Clear bache? I hope this is visible to you, right? Now you will see bache. First of all, we are classifying our organic compounds in two parts. What is two parts? First is open chain, or you can say acyclic, or you can say aliphatic compounds. And second, which one, bacha? Second is closed chain, or you can say ring compounds or cyclic compounds. Open chain means, if I give example, look at this. This compound is open chain or closed chain, beta? This is making an open chain or closed chain. Straight open chain, clearly, definitely, right? But here if I say, this is our what? This is our, this one, or I will say this one. These are open chain or closed chain? These are closed chain. These are closed chain and these are open chain. So we divide in two parts. We divide in two parts, closed chain and open chain. Very good. Very good. Now in open chain, if you go further, you will see, bache, look at this. Here I am having C, single bond, fine, C here. Now here I will see C, double bond C here, and here maybe I am having like triple bond. In open chain, if I go further, there are two parts, saturated compounds and second which one? Unsaturated. Yes, beta. In open chain, two possibilities are there, saturated and which one? Unsaturated. Saturated and unsaturated, right? Clear? Okay, fine. Clear, bacha? Right? It is saturated means only single bond. These compounds will have only single bonds, alkane only, alkane. There is no pi bond. Those compounds which have only carbon single bond, there is no double bond. What do we call them? These are saturated compound, right? And this one where pi bonds are present, maybe double bond, maybe triple bond. What do we call this? Unsaturated. We call this unsaturated, right? I'm saying once again, open chain have two parts. One is only single bond, only sigma. There is no pi bond in this type of compound. What are these? Saturated. And if pi bond comes between carbon, if between carbon pi bond is coming, what do we call them? It is unsaturated. Fine, very good. Okay. Now, bache, if you go to again closed chain, in a closed chain, if you see homocyclic, heterocyclic. Now see carefully, very carefully. If I make a compound like this, here carbon, carbon. In this chain, all are carbons only. In this chain, all are what? Carbons. So it is a homocyclic compound. When in this cycle, only carbons are present or only same atom are present, we call them what? Homocyclic, right? Whereas, look at this carefully. If I draw this here, I hope it is visible. Look at this carefully. In this case, beta, if you see, here we have carbon. But what is it? Nitrogen. If in this cycle, some other element also coming, like here we have carbons, and what is coming? One, nitrogen. So in this cycle, if some other element comes, we call this heterocyclic compounds. We call them heterocyclic. Clear, bache? Very good. Yes, bacha. So closed chain in two parts, homocyclic, heterocyclic. Homocyclic, heterocyclic. Very good, right? And next one, if you go, you can divide homocyclic in two parts. Some are known as alicyclic and some are what? Aromatic. This I will tell you in detail. Okay, you don't worry for now, because aromatic compound is a detailed topic. Okay, we will take it as a separate topic, separate class for this, right? So homocyclic, we go further in which beta? Alicyclic and aromatic. Aromatic compounds follow certain rules And if your compound is not aromatic, it will come in which category? Alicyclic. Okay, bache. These details you will see. We will understand in detail. Don't worry about it. Right? Clear? Okay, bache. Fine, fine, fine. So I'm saying again two parts. Organic in two parts. Open, close. In open, saturated and unsaturated. In close, homo, hetero. Understand this much for now. Cyclic in two parts. Homo, hetero. Homo further will go into alicyclic, aromatic. This I will discuss in detail in some lecture. Clear, bache? Very good. So please give me answer now for these questions. Okay? Try these questions. Tell me it is open or what? Open, close, saturated, answer this way. Okay? Please answer. Question is, classify the following. Right? So first bit I am giving here. Carefully give the answer. CH3, CH2, CH2, CH, CH3, CH3. Please tell me, it is open or close? If it is open, then which type? Saturated or unsaturated? Very good. Yes. So in our category, it is which one, beta? Open chain. It is open chain. Very right. It is open chain. In open, which type? In open, which type? Saturated or unsaturated? In open, it is which type? Saturated or unsaturated? Tell me, beta. Very good. There is no pi bond, only single bond. What is it? It is saturated. Right? It is saturated. Very good. Clear? Fine, fine. Now, second example I am asking. You are having here CH3, CH double bond, CH2 or CH you can say here, CH3. Now tell me for this. Fast, fast. Open or close? Open or close? Yes. It will be which one? It's open. It's open chain compound. Next one, which one? It is unsaturated. Because what it have? It is having pi bonds. It is having pi bonds. Very good. Very much clear. Fine, fine. Very good, very good. Okay, third one. Is the third one I am saying. Again, very simple. CH3C, triple bond C, CH2, CH3. Now tell me. Now tell me. Very good. Yes, again you say open chain. It is open chain. It is also which one? Unsaturated. Right? Tell me, which one is alkane, which one is alkene, which one is alkyne? Out of 1, 2, 3, which one is alkane, which one is alkene, which one is alkyne? From your 10th class only, nothing new. Yes, beta, this is what? This is our alkane. This is alkane, right? This is alkane. Second one is what? This is alkene. This is alkene, fine. And third one is what? Alkyne. Third one is alkyne. Very good. It is alkyne. Fine, fine. You are doing very well. Very good. Okay, okay. Next. Next I am asking. Fourth one, please tell me. Fourth one. Okay, tell me the fourth one. This one is what? This one is cyclic or open? It is cyclic. Cyclic, or you can say closed chain, right? Cyclic or closed chain, both are fine. Very good. Cyclic. Next, tell me, homo or hetero? It is homo or hetero? Homo only, because there is no other atom, only carbon, 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 carbon. Right? Very good. Next thing is, it is saturated or unsaturated? cyclic, homo, saturated or unsaturated? Very good. This is unsaturated, right? Very good. Very good. Right. Very right. Clear? Okay, next I am asking one more question. Give the right answer. Next thing is, or I will say oxygen. We have oxygen here, right? Tell me what it is. Very fast, very fast. Yes, it is which one? Cyclic. It is cyclic. And which one? Cyclic. Next one. Hetero. Right? It is cyclic and hetero. Cyclic and hetero. Right? Very good. Very good. It is aromatic or not? That thing we will discuss in some separate class. Okay? I am just giving one example to you. Look at this. Now, for now, you cannot find it is aromatic or not. It's a detailed topic, but I am just giving one example. This one. It is example which is what? Cyclic. It is cyclic, homo, and it is aromatic also. For aromatic compounds, we have a separate class. Don't worry about it. Okay? This I will tell you later. For you now, cyclic and which one? Homo. Cyclic and homo. That's it. It's enough for you. Remaining things we will discuss. Okay? Okay, everyone. I hope you are clear about this thing. Fine. Aromatic I will discuss. Nilaveni, aromatic I will discuss in separate class. Okay? It is a different detailed topic. Fine, bacha. Clear? Now, classify these, some of the compound. Tell me. Okay? Classify the following, bacha. Just try to classify. And imagine this as carbon. Take it as carbon only. This is carbon. This is also carbon. And this is carbon. Classify the following. Classify the following. Fast, very fast. Classify the following. What is it? Open chain. It's better is what? Open chain, first one. It's open chain, very right. It's open chain, right? And definitely it is open chain, simple beta. Alkane I will say, or I will say saturated. Fine. Very good. This one. Again I will say same thing. It is also open chain. And what? Saturated. Clear? Very good. Now tell me this one. This one, please. Okay, I am saying it is carbon. Take it as carbon, okay? Carbon. What is it? Yes, it is which one? Open chain. It's open chain and which one? Unsaturated. Because it have what? Pi bond. Unsaturated and it is having pi bond. Very good. Next thing. Again same beta. Open chain. What is it again? As it have pi bond between carbon, it is what? Unsaturated. Very good. This one. Fast. So this one beta, fast. This one is again. It is cyclic compound. Cyclic. In cyclic which one? Homo or hetero? In cyclic, homo. Very good. Again this is very right. Cyclic and which one? Homo. It is homo. Right? And homo I am saying, and which one? Saturated, because there is no pi bond. Now this one. It is cyclic. Definitely homo. And special thing, it have pi bond. It is what? Unsaturated. Because it have carbon pi bond. Carbon pi bond. Very good. And next question we just did now, few minutes back, it is what? Again, it is cyclic, but which one? Heterocyclic. It is heterocyclic. It is heterocyclic. Fine, bacha? That's it. For now, this classification is enough for us. Remaining we'll see. Okay, bacha? Aliphatic, aromatic, we'll see these things. Okay, bacha? Benzenoid, non-benzenoid, all these things will come. Okay, when we will study the topic aromatic compound. Okay, bacha? Fine. Okay, now I am starting next topic. Okay. Okay, bache. So, thing is degree of carbon, right? I'm asking here degree of carbon. What I mean by this, you will see, sometime we call it, it's a primary carbon. These words you will see many times. Primary carbon. Or sometime you will see, I will use word, it is secondary carbon. Okay. Sometime you will see, we'll call it tertiary carbon. So what this means, we have to understand, right? And D also possible, we can say we are having quaternary carbon. Okay, so what these means, beta? Okay, so carefully try to understand. Okay, right. So first I will say primary carbon, beta, see carefully. This is our carbon, okay, and this carbon is connected to, carefully give the answer. Okay, or I am making it more simple. Think about this carbon. This carbon is connected to how many other carbons? Only one, na? A carbon which is connected to only one more carbon. Or I will say a carbon which is connected to the one more carbon, which one? Primary carbon, right? This carbon, I will say which carbon? It is primary carbon. Because it is connected to only one more carbon. Remaining three are what? Hydrogen. Okay, just to elaborate this, I am showing this way. Imagine this carbon, hydrogen, hydrogen, hydrogen. And what it is? CH3. So this our carbon, this carbon is connected to three to hydrogen and one only what? One carbon. This is primary carbon. What do we call this, beta? This is our primary carbon. Clear? Very right. Secondary carbon, see carefully. See it carefully. We have here, assume CH3, CH2. What is it? CH3. A carbon which is connected to two carbons on two sides. I will say two sides. Like here I am showing, bacha. See carefully. This is a carbon. Here I have hydrogen. Here I have hydrogen. Here I have something what? CH3 something, CH3 something, right? So this carbon is connected to two hydrogens or something else and connected to two carbons, right? This carbon is which one? Secondary carbon. This carbon is secondary. Clear, bache? So a carbon connected to two more carbons this way, one bond with here with carbon and one here, it is secondary carbon, right? It was connected to only one more carbon, so it was primary. Clear, bacha? Very right, right. Now see carefully, next one I am saying, imagine this carbon is here, this carbon is connected to something like this, CH2, CH3 here, assume. Here we have CH3. Here I am saying, here we are having, assume CH2, CH3, something like that, and here is hydrogen. So better tell me carefully, this carbon is connected to how many carbons? This carbon is connected to how many carbons? 1, 2, 3. So this carbon is connected to 3 carbons. That's why it is what? Tertiary carbon. This is tertiary carbon. Very good. Tertiary. So our carbon is connected to how many carbons? It will decide it is primary, secondary or tertiary. Very good. Very right. Very right. Clear? And next one I will say, here bache, look at this. This is what? This is a carbon here. CH3. Here CH3. Imagine something else. I am saying CH2, CH3. Something else. You can say CH2, something like Cl or something else, right? This carbon is what? This carbon is connected to 4 more carbons. This carbon is connected to 4 more carbon. That's why what do you call it? It's a quaternary carbon, you can say. It's a quaternary carbon. Very good. If our carbon connected to 1 more carbon, primary. Our carbon connected to 2 carbons, secondary. 3 carbons, tertiary. 4 carbons, which one? Quaternary. Very right. Very right. Okay. Clear. I hope you are clear. Fine, bacha? Okay. So now I am giving question on this basis. I hope you have done up to here. So please finish it fast. Is bacha, finish it fast, fast. Fine. Now see bacha carefully. Look at this. Question is, find number of primary, secondary, tertiary and quaternary carbons in given molecule, right? Okay, bache, find it. Here we have CH3, CH2, CH3, CH2, CH3 beta CH3, CH3, right? Yes, everyone. In this molecule, find how many are primary carbon, how many are secondary, how many tertiary. Look at the carbon carefully and give me answer. Look at all of these carbons very carefully, give me answer. First, think about this one. This carbon. This carbon is primary, secondary, tertiary. This carbon is primary carbon because it is connected to 1 more carbon, right? This carbon is what? This carbon is secondary carbon. Why? Because connected to 1 carbon here, 1 carbon here, right? This carbon beta is which one? Tertiary carbon. Why? Connected to 1 carbon, 1 carbon, 1 carbon, right? This one is what? Secondary. How secondary? Connected to 1 and 1, 2 carbons, right? This one quaternary, connected to 1, 2, 3, 4. Clear? And what is this? It is again primary because only 1 connection. It is also what? Primary, 1 connection. It is also what? Primary, 1 connection, right? So in complete molecule, how many primary are there? Tell me. How many primary carbons are present? Tell me, bache. Fast, fast. In complete molecule, how many primary? 1, 2, 3, 4. Total 4 primary carbons. How many secondary carbon are present here? How many secondary carbon? Secondary 1, 2. Right? Sorry. How many are tertiary carbon? How many tertiary carbons? Tertiary carbon. How many? 1, right? Only 1 is tertiary. Fine. And how many quaternary, bache? How many quaternary carbon? Answer fast, fast, fast. 1. Fine, 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 fine. Oh, wait, beta, wait. See carefully, this carbon we didn't check, na? So please check this. Here, this carbon is missed, right? It is present here. 1 more carbon, right? Total how many now? So 1, 2, 3, 4 and 5. Primary are 5 here. We missed this one, right? We had to see this one also, na? This was uncounted. Okay, done, bacha? Same question I am giving one more. So please answer this carefully. Same question I am giving. Answer carefully, bache. CH3, C, CH2, CH3. CH, CH3, CH3, right? CH2, CH, CH3, CH3, right? Same question beta. Same question. Count total. Give every carbon primary, secondary, tertiary category and then tell me how many are present. Okay? Very good. Fast. First of all, give every carbon its degree and then count total. Then count total also. Fast. Yes, everyone? Yes. Count. Look, bacha, this carbon primary, secondary, what? Tell me. This carbon primary because connected to one carbon only. This carbon is what? Tell me fast. 1, 2, 3, 4 connected to 4 carbons. So it is what? Quaternary. This carbon connected to how many carbons? 1, 2. Which one? Secondary. This one primary. This carbon connected to how many carbon? 1, 2, 3. Which one? Tertiary. This carbon here, 1 carbon, 2 carbon, so it is what? Secondary. This carbon, 1, 2, 3, it is what? Tertiary. This carbon, primary. This carbon, primary. Clear, bacha? Okay, done. Now count. Total how many primary carbons are present here? Count, beta. Wait. This carbon also what? Primary. This carbon also what? Primary. So carefully give the answer. There are many carbons, okay? There are many carbons, right? How many primary? 6. Many are saying 6 answers. Check carefully, right? Primary 1, 2, 3, 4, 5, 6. Very good. 6 are primary. Count beta secondary carbons. Fast. Count secondary. Secondary carbons how many? Secondary how many? Secondary how many? Secondary beta. Very secondary. 1 secondary. 2 secondary, right? There are 2 secondary carbons. Okay, fine. Okay. Tertiary carbon. Tertiary carbon how many, bacha? Find fast. Tertiary. Which is tertiary? 1 tertiary. Tertiary, right? So 2 are tertiary. Fine, bacha. And how many are quaternary carbons? How many are quaternary carbons? Very right. Which one? This one. 1. Okay. Yes. You can count easily, but you should understand how to check it is primary carbon or secondary carbon or tertiary or quaternary. Fine. The counting that you can do easily. There is no doubt. Counting you can do easily, right? I am saying once again, if our carbon connected to one more carbon only, it is primary. Connected to 2 more carbons, connected to 1 only, primary. Connected to 2 means it is what? Secondary. Connected to 3, tertiary. Connected to 4, quaternary. Fine. Clear. No, you cannot put methane in this category. It is separate. In methane, carbon is not connected to any other carbon, right? So it's a different category. You can't say it primary, secondary, tertiary. Okay, bacha, you got your answer. Venkat, you can't call it primary, secondary, tertiary. In methane only we have what? In methane, beta, we have only hydrogens, na? This carbon is not connected to any other carbon, only hydrogens. So you cannot say primary, secondary, tertiary. Okay? Clear, everyone? I hope we are clear. Degree of amines, degree of alcohols, degree of halides we'll see tomorrow. And then we will start our nomenclature. Okay? Step by step. Okay, bacha? So see you tomorrow. I hope this will help you. So read this from your NCERT and we will start nomenclature tomorrow. Okay, bacha? Bye then. See you. Bye. And don't forget to like and subscribe, right? and don't forget to fill the form in description. Okay बच्चे, bye, see you.
Students, good morning. Good morning, Ra. Today's our botany topic is changes in a flower after fertilization. Changes in a flower after fertilization, also called post-fertilization structures and events. Simply you can say post-fertilization changes in ovary of the flower. Post-fertilization structures and events, it is the topic name. Mainly three things occurs after fertilization in a flower. One is the development of endosperm. Event number one is the development of endosperm. Second event is the development of an embryo. Second event is development of an embryo. Event number three is maturation of ovule, nothing but development of ovule into seed. That is the third event. Fourth and last event is maturation or ripening of a ovary into fruit. Tell me, how many events are there after fertilization in a flower? Four. Four are the four events of post-fertilization. Number one, endosperm formation. Yes. Number two, embryo formation. Number three, seed formation from a ovule. Number four, fruit formation from ovary. Events of post-fertilization. Number one, formation of endosperm. Second, embryo formation. Third, seed from ovule. Fourth one, formation of fruit from matured or fertilized ovary. These are the four important events occurs after fertilization. What kind of fertilization is found in flowering plants or angiosperms? For the name of that specific kind of fertilization, double fertilization. Double fertilization is the most important event and characteristic feature of angiospermic plants. In the double fertilization, from male parent or male pod, first sperm cell is fused with egg. Egg is the contribution from female organ or maternal, male is the paternal. Ploidy of the sperm is haploid, ploidy of the egg is also haploid. Fusion of these two gametes, egg is the female gamete, sperm is the male gamete, fusion of these two types of gametes is known as. Now, what is the meaning for this kind of gametic fusion? Syngamy. Syngamy. Fusion of two gametes is the syngamy. Result of the syngamy, zygote. What is the ploidy of the zygote? Diploid cell. Syngamy. Second kind of gametic fusion. Second one is within a single pollen grain, two sperms are there. Already one sperm engaged with egg. What about the second sperm? It engaged with the central cell. Secondary nucleus found in central cell and give rise to triploid structure. The name of that triploid structure is the pen. What is the full form of the pen? Full form of pen, primary endosperm nucleus. This kind of fusion is called triple fusion. Okay, this kind of gametic fusion is the triple fusion. Triple means three types of sets of the chromosomes. Two sets of the chromosomes from central cell, one set of the chromosome from sperm involved in fusion, hence it is called triple fusion. The final result is the primary endosperm nucleus. Yes, now it is the topic here. Listen carefully. This 2 minutes class is very important. Be alert. Among these two structures, zygote and pen, which one is the bigger or which one is the elder one? It means the first formed one. Zygote is the first formed one or pen is the first formed one? Zygote is the first formed structure. Elder, pen is the younger. Later, zygote give rise to an embryo. What is the first structure for the development of a diploid embryo? Zygote. Likewise, what kind of structure is obtained from further development in this pen? Due to the further mitotic cell divisions in primary endosperm nucleus, a kind of product is obtained. What is the name of this product? What is the name of product obtained by the activity in pen? Endosperm. Endosperm. What is the difference between embryo and endosperm? Usually embryos are diploid, but endosperm is in triploid. The ploidy of endosperm is the triploid, ploidy of embryo is diploid. Because the embryo obtained from diploid structure, that's why it is also in diploid condition. The first cell for the development of an endosperm is the pen. Pen ploidy, 3N, that's why endosperm is also in triploid condition. Here one important thing, zygote is formed earlier than pen. After that, zygote undergo some time and resting period. Meanwhile, pen undergo repeated number of divisions to form endosperm. So, endosperm formation is always earlier than that of an embryo. Within these two structures, zygote is formed earlier than pen. Whereas in these two structures, endosperm formation is earlier than that of embryo formation. Why? Endosperm formation always earlier than embryo. For development of embryo, food matter is required. The food supplying tissue for the development of an embryo from zygote is endosperm. Tell me once again, what is the functioning of endosperm? The functioning of endosperm, it provides nourishment or nutrition for development of an embryo from zygote. To develop an embryo from zygote, food is required. That food supplier is the endosperm. It is a need or necessity for the development of an embryo from zygote. Endosperm always provide nourishment for the development of an embryo from zygote. Yes, nourishment or nutrition supplying. It is the reason for earlier formation of endosperm than that of embryo. And one more important thing, identify the structure. Guess what is the structure? Embryo sac after fertilization. Towards the chalazal side, 3 antipodals already degenerates. 2 synergids present either sides of egg already degenerates. 3 antipodals degenerates, including nucleus. Synergid also degenerates. Only 2 cells remain. Fertilized egg is the zygote, and fertilized central cell is the pen. Or the fertilization, this is the 7-celled embryo sac become as 2-celled structure only. One is the diploid zygote, another one is the triploid pen. Only these two are retained, remaining all are degenerates. How many cells are degenerates? 3 antipodals, 2 synergids, 3 plus 2 total 5 cells out of 7 degenerates. Okay. Zygote takes some resting period or hibernation period. Meanwhile, pen undergo repeated number of free nuclear divisions. The number of nuclei multiplied. All these are cells daughter nucleus of primary endosperm nucleus. Due to the repeated number of free nuclear kind of mitotic cell divisions in pen. Later, cellularization occurs, means formation of cell wall around the nuclei. Now, this entire diploid tissue is called as endosperm. It is the mechanism for formation of endosperm. It is the mechanism for endosperm formation. Endosperm. Whenever endosperm is started, then only zygote undergo divisions to form embryo formation. For the development of an embryo from zygote, food is required. That food supplier endosperm formation should be earlier than that of embryo formation. Due to the repeated number of divisions in zygote, gradually embryo size enhanced, then amount of endosperm completely depletes. As increase in size of embryo, the quantity of endosperm declines. Quantity of endosperm declines. I will draw two diagrams. Identify the variation between these two diagrams. Diagram 1 or one embryo sac, second embryo sac. It is with zygotes, Z for zygote. Here also zygote is there. A, B. Tell me what is the difference between diagram A to the diagram B? Identify the difference. In the diagram A, cellularization started. Cellularization means conversion of nucleus into cell by forming a cell wall around each nucleus. Around each nucleus, cell wall is formed. It is the meaning for conversion of nucleus into cells. The pink color structures present inside of the embryo sac are nucleus. The green color structures are cells. So, endosperm is present in two forms, nuclear form and cellular form. Tell me what is the difference between the two diagrams A and B. In the diagram A, purely nuclear form of endosperm is there. Nuclear form of endosperm. But here both the forms are there. At peripheral area, cellular form. Cellular form of endosperm. Central nucleated condition is also there. In diagram A, endosperm is present in two forms, cellular form and nuclear form. The central area is the nuclear form. Peripheral or greenish area is the cellular form. Whereas in diagram B or embryo sac B, purely nuclear form. The first form of the endosperm is the nuclear form. Gradually, that nuclear form of endosperm turn into cellular form. The cellularization phenomena means formation of cells starts from periphery to center. Periphery to center. What is the morphological nature of the fruit or coconut water? What is the morphological nature of water present in coconut? Coconut water is an example for purely nuclear form of endosperm. While you taking coconut water as the edible material, you should feel each and every drop of the water is a group of nucleus of endosperm. Okay, coconut water is the best example for nuclear form of endosperm. This is the nuclear form of endosperm found in tender coconuts. Tender coconuts. At the time of the maturation or ripening of that coconut fruit, gradually some little amount of that coconut water turn into kernel or copra. In Telugu, kobbari. Copra and kernel. that kernel is or copra is cellular form of endosperm. Here, liquid form of endosperm turned into solid form. Liquid coconut water is an example for liquid endosperm. Technically, technically coconut water is or liquid form of endosperm is nuclear form. At the time of the maturation, the liquid form turned into solid. Solid one is the copra or kernel, coconut kernel. It is the meaning for conversion of nuclear form of endosperm into solid form or liquid form into solid form or nuclear form into cellular form. Okay, give the example for nuclear form of endosperm, coconut water or what is coconut water? What is coconut water? Nuclear form of water or nuclear form of endosperm or liquid form of endosperm. What is the difference between coconut water to the copra? Telugu name of the cobra, kobare. What is the difference between coconut water to solid form of endosperm? It is the cellular. Copra is the cellular. Coconut water is the nucleus. Gradually, such nucleus are modified into or become as a cells. That process proceeds from periphery to center. The process proceeds from periphery to center. That's why coconut water is found at the center, peripheral area, this copra is there. Copra. What is the main need for formation of this kind of endosperm? For development of an embryo from zygote. Development of an embryo from zygote, food is required. I will explain one more concept here. Zygote gradually turns into development of an embryo. It is an embryo already developed from zygote. Even some little amount of endosperm remains here. From as time passes on, size of embryo enhanced. Embryo becomes as a very bigger structure and fully matured structure. This entire white color portion is an embryo. But a little amount of endosperm may remains. Remaining entire endosperm is utilized by embryo. As increasing size of the embryo, endosperm quantity declined. The quantity of endosperm declined, but size of the embryo enhanced. It clearly explains embryo consumes endosperm. So endosperm is the food for embryo. Now identify this diagram. Whereas in the diagram, total area of this embryo sac or seed is occupied by embryo only. There is no endosperm because the entire amount of the endosperm utilized by this embryo. This kind of two types of development occurs in seeds. Fertilized ovules are called seeds. The seeds are broadly classified into two categories. One is the endospermic seed. Another one is the non-endospermic seeds. Seeds two types based on presence and absence of endosperm. Tell me once again, what is the difference between endospermic seeds and non-endospermic seeds? Think, concentrate. What is the difference between endospermic to non-endospermic? Even fertilization, even after completion of the fertilization or development of an embryo, if some amount of endosperm is remain, such are called endospermic. If the endosperm is absent, because total amount of endosperm already consumed, utilized by this embryo, such are called non-endospermic seeds. So the major difference between endospermic to non-endospermic seeds, total amount of endosperm is utilized in non-endospermic. That's why endosperm is absent. Whereas in endospermic seeds, some amount of endosperm is retained. Due to the presence of this endosperm, the name of the seed is endospermic. The name of that seed is endospermic. Once give the definition for endospermic, the seed with endosperm. Endosperm formation is mandatory, but some amount of endosperm is left over even completion of development of an embryo. Whereas in non-endospermic, here also endosperm formation is there, but that endosperm completely depletes or utilized by this embryo. So endosperm formation is common in both the seeds. But here endosperm is completely consumed or utilized, but is some amount of retained in endospermic seeds. It is one of the variation between endospermic to non-endospermic seeds. After fertilization, these are the seeds are formed from ovules. Seeds fertilized. After fertilization, ovary become as a fruit. Fruit from ovaries, seed from ovules. Fruit is the characteristic feature of flowering plants. In the entire plant kingdom, only flowering plants fruits are present. No fruits in algae, fungi, bryophyta, pteridophyta, even in gymnosperms also fruits absent. Development of fruit, presence of fruit, formation of fruit is the characteristic feature of angiosperms only or flowering plants only. Definition for fruit is matured ovary, fruit, ripened ovary, fruit, developed ovary, fruit after fertilization. Each and every one is the fruit. Even rice, paddy grain, maize grain, almonds, chilies, tomatoes, bitter gourd, cucumbers, bottle gourd, snake gourd, rigid gourd, everyone is a fruit. Vegetable fruits such are. There is also normal edible fruits. Sometimes without fertilization also ovary become as a fruit. Fruit formation without fertilization. Formation of fruit without fertilization is known as parthenocarpic fruit. Once give the definition for the parthenocarpic fruit. Formation of fruit without fertilization. Even absence of the fertilization, fruit formation is there. That is the meaning for parthenocarpic fruit. Give an example for parthenocarpic fruit. Give an example for parthenocarpic fruit. Okay, yes, right. Parthenocarpic fruits are generally seedless. Seedless grape and seedless bananas are very good example for parthenocarpic fruits. What is the reason for absence of the seeds in parthenocarpic fruits like watermelons, bananas and grapes? Lack of fertilization. Yes, such a reason question. Seeds are absent in parthenocarpic fruits. What is the reason for lack of seeds in parthenocarpic fruits? Absence of the fertilization. Is also one of the best area for the construction of assertion reason. This is a parthenocarpy two types. Natural parthenocarpy and artificial parthenocarpy. By using gibberellin hormones, by using gibberellin hormone or GA, gibberellic acid, we can induce artificial parthenocarpy. Even in fully ripened banana, at the center place black color dots are present. Such are unfertilized ovule, such are not yet seeds. Thus, aborted ovules or unfertilized ovules appears a black color structures at the center of the fruit. Okay. Banana is the example for parthenocarpy. Tell me once again what is the reason for absence of fruits in banana or parthenocarpic fruits? Lack of fertilization. Ovules unable to turn into seed. Definition for the parthenocarpic fruit forms without fertilization. Parthenocarpy. This phenomenon is called parthenogenesis. Parthenogenesis. Name the two parts of a seed. The fruit is whatever it may be. In entire fruit, mainly two parts are there. One is the wall, another one is the seed. In entire fruit, only two parts are there. Tell me once again. Name the two parts of a fruit. Hardest part of the fruit is the seed. Second part is the wall. The technical name of wall is the pericarp. The technical name of fruit wall is the pericarp. The pericarp may be dry or fleshy. In fruits like tomato, banana, apples, grapes, oranges, pericarp is the fleshy. In the fruits like almonds, pericarp is the dry. So that's why such fruits are called dry fruits. Kaju, almond, pistas. So fruit wall pericarp is the dry. If the pericarp is the dry, such fruits are dry fruits. If the pericarp is the fleshy or pulpy, like watermelon, fleshy fruits. Observe apple. In apple, this entire edible portion is the pericarp. And the central seeds are present. So each fruit consisting of only two constituents. One is the seed, another one is the pericarp. The common name of this pericarp is the fruit wall. The common name of the fruit wall is the pericarp. The pericarp is the fleshy, whereas here pericarp is a dry. Due to the presence of dry pericarp fruit wall, these fruits are called dry fruits. Due to the presence of fleshy pericarp or fruit wall, these the fruits are an example for fleshy fruits. In most of the plants, the fruits develop from ovary. Fruit develop from ovary. Sometimes other floral parts also degenerates, and generally other floral parts of degenerates or fall off. Observe some pictures. Thalamus of the flower. Ovary is placed exactly at the top area of the thalamus in most of the flowers. The carpel possesses ovary style stigma. Along with this carpel, stamens, petals, and sepals also present. After the fertilization, only this ovary becomes as a fruit and ovules into seeds. Remaining all floral leaves, sepals fall down, petals fall down, stamens fall down, and style and stigma also withered. Style and stigma withered. Only ovary is the persistent structure. Sometimes along with the ovary, other floral parts like thalamus, and pedicel is also involved in formation of fruit. If the thalamus or pedicel in fruit formation, such kind of fruits are called false fruits. Based on development, fruits two types. Fruits are two types: true fruits and false fruit. If any kind of fruit is obtained from only ovary, true fruit. If the fruit developed from other floral parts like thalamus and pedicel, such are called false fruits. Once give the definition for both true fruits and false fruits. If the fruit developed from only ovary, that is a true fruit. If a fruit developed from other floral parts like thalamus and pedicel, including ovary, such are called false fruits. Here one question: name the plant organs or organs of the flower involved in false fruits formation. Thalamus and pedicel. Thalamus or the pedicel also contributes for the fruit formation. Such fruits are called as false fruits. Such fruits are called false fruits. Now guess what is the name of this fruit? Identify what is the name of this fruit. Cashew nut fruit. Cashew nut. In cashew nut, in the fruit cashew nut, it is the pedicel. Pedicel appears like a fleshy fruit. The true ovary become as a true fruit. It is the real ovary. Ovary become as a true fruit. In this plant, cashew nut plant, two types of fruits are there. One is the false fruit, another one is true fruit. Both the fruits are side by side. True fruit develop from ovary, false fruit develop from pedicel. The name of this plant is the cashew nut. Cashew. In the real edible portion is the true fruit, cashew. Sometimes somebody may eat false fruit is also as a edible part. Okay. In apple, this entire edible portion of the apple is the false fruit and is the thalamus. Thalamus is the only edible part in apple. After eating of this entire fleshy thalamus part, one central non-edible portion remains, that is the true fruit. So true fruit in apple is non-edible. Thalamus is the edible. Whereas in cashew nut, both the false fruit pedicel, true fruit ovary, both are edible. Tell me once again, give the examples for false fruits, cashew nut and apple. Cashew nut, apple, both are example for false fruits. What are the fleshy parts and edible parts in apple plant or apple fruits? What is the edible portion? Thalamus. Non-edible portion is the centrally located, non-tastiest hard part is the true fruit, that is non-edible. Whereas in cashew nut, false fruit develop from, one question ready here. In cashew nut, false fruit is developed from pedicel. What are the edible parts in cashew nut? Both true fruit, false fruit. Likewise, one more question frequently asked in previous years, that is what is the edible portion of the apple? Or what is the eatable part of the apple? Thalamus. Thalamus involved in formation of false fruit. Centrally located portion of this fruit and seed contained portion is the true fruit. So seeds are present in false fruits or true fruit. In true fruit only seed is present. Strawberry is also another example for false fruit. This red color portion, fleshy portion, edible part is the false fruit developed from thalamus. Centrally hardest part is there, that is the true fruit, but non-edible. No. Somebody asked, Ahalya, thalamus is the edible portion in apple and strawberry. Thalamus is edible, not a non-edible portion. Okay, but the true fruit found inside of the apple, central part of the apple, that is the non-edible portion. Answer for this question: which part of the flower contributes for the formation of false fruits? Name the part of flower involved in false fruit formation. Thalamus and pedicels. Name the plant in which thalamus contributes for false fruit formation. Thalamus, apple, and strawberry. Pedicel, cashew nut. True fruit develop from fertilized ovary. Ovules develops into seeds and ovary into fruit. Guess what is it? Mango. In mango, the central portion, not visible in this image, inside of this area, one seed is there. Outermost part or greenish or yellowish thin layer is called epicarp. Middle, the juicy part is mesocarp. Inner hard and stony part is endocarp. These three are the three layers of pericarp. These three are the three layers of pericarp. In the fruits like mango, pericarp differentiable into three layers. Name the such layers. Outermost layer of the pericarp is epicarp, middle layer is mesocarp, innermost layer is endocarp. Now the question is here, what are the edible portions of mango fruit? What is the edible portion of mango? Both epicarp and mesocarp. In case of a mango variety rasalo, only mesocarp is edible. Whereas in other kind of varieties, both mesocarp and epicarp are edible. But endocarp is non-edible. By breaking that endocarp, you can observe one seed. So seed located inside of endocarp in the fruit mango. These two layers are fleshy, hence mango is an example for fleshy fruits. What is this coconut fruit? This hardest stony area present around the coconut fruit is the endocarp. Endocarp. That white color portion, guess, give the name for this white color portion of the coconut fruit, that is edible portion. Botanical name, or sorry, the technical name of this white color portion. When this is the coconut fruit is the tender, at the center water is there. At the center water. Peripheral area, this white is solid portion is there. The name of that solid part, solid or cellular endosperm. Recently have learned cellular endosperm. Central, so water is remains, that is the nuclear form of endosperm. Nuclear endosperm. I will ask some questions, ready to answer. What is the edible part in mango? Epi and meso. Likewise, what are the edible part in coconut? Only endosperm is the edible part. That endosperm available in two forms. Solid form is the cellular, liquid form of endosperm, that the coconut water is nuclear endosperm. Coconut water is nucleus endosperm. Endosperm. While eating this copra, you should remember that word endosperm. While eating mangoes, you should recollect or remember epicarp and mesocarp portion is edible. Try to answer for these questions. Fertilized ovary is called. Fertilized ovary. Fruit. Fourth option. Second question. Parthenocarpic fruit is formed. Parthenocarpic fruit is formed. Okay, good. Answer for next question. Parthenocarpic is formed without fertilization or with fertilization, before fertilization, none of the above. Yes, without fertilization of ovary. Without fertilization. Next question. Example for parthenocarpic fruits. Example for parthenocarpic. Mango or cashew or banana or apple. Tell me. Banana. Seedless fruits. Parthenocarpic. What about apple? Apple and cashew both are example for false fruits. Cashew, apples are false fruits. Banana is parthenocarpic. Why banana is an example for parthenocarpic fruit? If the fruit formation occurs without fertilization. How can you say both cashew and apples are example for false fruits? Here, other floral parts like pedicel and thalamus involved in fruit formation. That's why the fruits in apple and cashew are good example for false fruits. Okay, same question. False fruits are formed from thalamus and pedicel. Name the plant in which thalamus involved in false fruit formation. Apple. False fruit from pedicel is found in formation of false fruit from pedicel. Cashew nuts. Thalamus and pedicel. Now, fertilized ovules or seeds. In each seed, mainly two parts are there. One is the seed coat, another one is an embryo. Earlier you learned one concept. Name the two parts of a fruit. In case of fruit, what are the two constituents of that fruit? Pericarp and seed. Pericarp and seed are the two parts of a fruit. Whereas in seed, name the two important parts. Seed coat and embryo. Tell me once again. Pericarp is the part of fruit or part of seed? Yes, pericarp is the part of fruit, but seed coat or embryo is the part of seed. The embryo is made up of an embryonal axis and either one or two cotyledons. Observe here. In each seed, especially in the plants like dicots, two bits are found in each seed. Mostly these two are similar in size and shape, such are called cotyledons. If the number of cotyledons are two, such plants are called dicots. Whereas in monocots, only one cotyledon or single cotyledon is there. Tell me once again, how can you differentiate the monocot seeds from the dicot seeds? If the number of cotyledons are two, dicot. If the number of cotyledons is only one, monocot. Cotyledon is a part of an embryo. Try to understand the structure of the seed. In seed, basically mainly two parts are there. One is the peripheral area is the seed coat. Entire remnant portion is called an embryo. This embryo is differentiable into two parts. One is the either one or two cotyledon and embryonal axis. In fruit, two parts, pericarp and seed. In seed also two parts, seed coat and embryo. Within embryo, two parts are there, cotyledons and embryonal axis. I will write once again on this board. Fruit consisting of two parts. Try to fill these blanks. What are the two constituents of a fruit? Pericarp and seed. Maybe single or more than one. Name the two parts of a seed. Outermost part of the seed is the seed coat. Another part is embryo. What are the two constituents of an embryo? One is the embryonal axis. Another one cotyledons. Number of cotyledons are differ from plant category to category. Seed is a answer for this question. Seed is a part of fruits. Embryo is a part of seed. Embryonal axis is a part of an embryo, and cotyledon is a part of an embryo. Note down. This is the most valuable information. By learning this table, you can solve for several questions here. Simplest question. This kind of question repeatedly asked in previous NEET examination. Cotyledon is a part of. First option fruit, second option pericarp, third of seed, fourth of embryo. Answer for this question. Cotyledon is a part of embryo only, it is not a part of seed, not the part of fruit. Likewise, seed coat is a part of seed. Embryonal axis is a part of an embryo. Okay, the total chapter sexual reproduction in flowering plant completed. Next our topic is the biomolecules. I will start after this class. Okay, students, bye.
Hello students. Hello everyone. How are you all? Hello. Good morning. Good morning everyone. Good morning Rajiv, Charvik, Deeptika, Butterfly, Malika Arjun. Good morning beta. How are you? I am fine beta. Pujita, Jayashri, Balu, Veda, Rajiv, Rohan, Jayashri. All right everyone. I hope you all are fine. I cannot take all the names. I hope you understand that I cannot take all the names, but all of you, a very good morning to all of you. All right, so let's start today's class. So do you know today's topic? Yes, everyone. Do you know today's topic? Today we are going to, understand about the power. Do you know we have a power? Good morning everyone. Immunity, Venkat Shiva Kumari, yes, absolutely correct. Immune system, immunity. So we will talk about this power, right? We have a great power to fight. I'm not talking about that power with which we are fighting with our friends, with which we are fighting with our, classmates or our siblings, brothers and sisters, right? We should not fight. So we should not fight with anybody, but yes, we should fight with the pathogens, because pathogens are causing the disease to us, right? Pathogens are causing the disease to us. Yes, everyone. So I will tell you a brief introduction of the health and disease, and then mainly I will talk about the immunity in today's class, right? So let us start. There is a chapter beta, there is a chapter in the 12th class NCRT. There is a chapter in 12th class NCRT that is human health and disease. Human health and disease. Human health and disease. So you should know about, we should be worried about us. We are humans, right? And we obviously everybody should be healthy. There are some factors which are affecting our health, and there are some factors which can maintain our health, right? So we should always take care of our body, of our health. We should always eat healthy. We should always, clean our body, our clothes, everything what we are using. We should be clean. We should be, healthy. We should be hygienic. All these practices are very, very important to maintain our health. So first of all, what is the meaning of health? Health does not mean that simply we say that I don't have any disease, so that means I am healthy. No. Absence of disease, obviously, simply we say absence of disease is healthy. But when you are healthy physically, when you are healthy mentally, and when you are healthy socially, then only we will say that you are healthy. Okay. So health is defined as a state of complete physical, mental and social well-being, beta. Social well-being. Health is a state of complete physical, mental and social well-being. You should be healthy, in all the terms, in all the aspects, physically, mentally, socially. How you behave with others, that is also an aspect of health. All right. So now, don't talk in the chat section among yourselves. If I'm asking something, then you should respond. Otherwise, don't create nuisance in the class, beta. This is also not healthy. You are not healthy if you are talking about anything else, and you are not talking about the class. You are not talking about the relevant topics going into the class. You are talking among yourselves. Then why you have come into the class? You can talk to others anywhere else, but class is not the place. Class is not the right place to talk among yourselves. Class means only you and teacher. What teacher is telling you, you should hear that, you should understand that, you should learn that, right? And what are your doubts, you should ask. So don't create nuisance in my class. I will not tolerate nuisance in my class. Okay? Once you'll come here, then you will understand how strict I am. I'm very strict in my class. All right. So now let us continue. Health. So I hope you should show me that you are healthy. If you won't talk and create nuisance in the class, then only I will teach further. Okay? And then only you will show me that you are healthy. So now show me in the class that you are healthy. You are talking about the things I'm teaching you here. Okay? Okay, everyone? Am I understood? Yes or no? Tell me in the chat section by giving a thumbs up. Yes, everyone. So shall we start? Shall we do a healthy start of the class? Yes? All right. Ma'am, don't be strict. I will be strict. Okay. Okay. Now coming to the disease. Disease means dis-ease. If you break this word, dis means absence. Ease means, ease means comfort. All right, everyone. So now let's focus. Dis-ease, beta, means discomfort, dis-ease or comfort. When you are not comfortable, when you are feeling something uneasy, when you are feeling uneasiness, discomfort, that means you have some problem in any of the organ. So when one or more organs are not functioning properly, when one or more organs are not functioning properly, that means you are not healthy. That means you have something called disease. All right? And do you know that diseases are caused by which organisms? What is the name of those agents which cause the disease? So beta, disease is caused by disease causing organisms are known as pathogens. Disease is caused by Diseases caused by pathogens. These pathogens can belong to any category. So many categories of pathogens are there, and that is why we have so many categories of diseases, beta. Alright, everyone is telling correct. Pathogen. Yes, pathogen. Pathogen means the disease causing organism. Okay, and this pathogen can be a bacteria, it can be a virus, it can be a fungi, beta. It can be a fungi, right? It can be a protozoa, right? It can be a helminth. So we will be studying helminthic diseases, protozoan diseases, fungal diseases, viral diseases, bacterial diseases. All right, we will be studying all these diseases in this chapter. But I am not talking about the diseases. Okay, I am talking about something which is very conceptual, and the understanding part of this chapter, the major understanding part of this chapter is beta immunity. Okay, so health you understood, diseases you understood. Now if we want to fight against these all pathogens, we have something called immunity. All right, so now I will be defining immunity. What is the immunity, beta? Immunity is the power. Immunity is the ability to fight against the disease causing organisms, or you can say that immunity is the ability to fight against pathogens. Okay, so immunity is the ability to fight against disease causing organisms, disease causing organisms. So yes, everyone, can you tell that this immunity is present in us by birth? Yes or no? Or we acquire it after birth? Everyone, yes, in the chat section. Now you should be active. When I am asking questions, you should be active. So is it present by birth? Yes, absolutely. It is present by birth. It is present in us by birth. We have this immunity. Why? Because we have a system that is known as immune system. So this immunity is provided by immune system, beta. This immunity is provided by which system of the body? Just like we have digestive system, we have respiratory system, we have nervous system, endocrine system, so many systems, circulatory system, excretory system. Just like that, we have immune system. All right. So this immunity is provided by which system, beta? This is provided by immune system. Now, what is this immune system? We know about digestive system, ma'am. Digestive system has this mouth, opens into buccal cavity, esophagus, stomach, intestine. Excretory system has kidneys, right? Inside the kidneys, nephrons are there. Nervous system has brain, spinal cord, right? So circulatory system has the heart, blood vessels, blood. So what is there in the immune system? What is making our immune system? All right, so everyone is talking about the colostrum. Yes, beta, yes, you know, all of you know all these things. Only I am just, giving a brief introduction. Okay, so you will get the details, the minute details. When you listen to the class, you will get the minute details. Colostrum, obviously, it provides you, you are getting it from the mother. So yes, colostrum has antibodies in it. We will be talking about colostrum. Don't worry. Okay, I will be telling you that the colostrum provides which type of immunity. That is why basics are required. Okay, so which type of immunity will be provided by colostrum? I will talk about it. But first of all, you should know what all things are making your immune system. Okay. What all things are making your immune system? So in your immune system, beta, there are lymphoid organs. The organs of immune system are known as lymphoid organs. Okay. There are two types of lymphoid organs. One will be the primary lymphoid organs, other will be the secondary lymphoid organs. Primary and secondary. Let me simply tell you, primary lymphoid organs are your bone marrow. You have a bone marrow. In the long bones mainly you will find the bone marrow where all the blood cells are getting formed. That bone marrow is the primary lymphoid organ. Okay, and one more lymphoid organ is primary, that is known as beta thymus. Thymus is present near your heart. Thymus is present near your heart. Okay, on the ventral side of the heart, this organ is present, which is also a part of the endocrine system. Thymus is also an endocrine gland. So you know about thymus, you know about bone marrow. These two things are the primary lymphoid organ. Now you will say, ma'am, what is the meaning of primary lymphoid organ? Okay, let me tell you the meaning of primary lymphoid organ. Primary lymphoid organs, beta. Primary lymphoid organ. One is bone marrow, another is thymus. One is bone marrow, another is thymus. Bone marrow is responsible for production of B and T lymphocytes. You know that very well, that all the blood cells are formed in bone marrow. So B and T lymphocytes are also the blood cells. They are WBCs. Yes or no? They are lymphocytes. Lymphocytes are type of WBCs. Yes or no, everyone? Do you know that? That is why, that is the reason I have taught you blood first. Because blood, what are the cells in the blood? They are coming everywhere. Okay. So now lymphocytes. Lymphocytes are also the type of WBC. WBC is a blood cell and blood cells are formed in the bone marrow. You know that. Now here only maturation of B lymphocyte will also occur. Maturation of B lymphocytes. So can we say that bone marrow is the site of production of B and T lymphocytes both, and bone marrow is the site of maturation of B lymphocytes, right? Bone marrow is the site of maturation of B lymphocyte. Maturation means what? Maturation means it will become function. It will become functional. It will do some function. When it is immature, it is not doing the function. When it is mature, it is doing the function. Okay, I will tell you what is the function. Now thymus. Thymus is the site of maturation of T lymphocytes, beta. Site of maturation of which lymphocytes? T lymphocytes. So it is based upon the maturation only. When B lymphocyte is matured in bone marrow, that is why it is starting with B. B means it is getting matured in bone marrow. Here T means it is getting matured in thymus. T for thymus, B for bone marrow. B lymphocytes are matured in bone marrow. T lymphocytes are matured in thymus. Have you understood this? Have you understood the function of primary lymphoid organ? Now coming to the secondary lymphoid organ. So when these B and T lymphocytes will be formed and matured in primary lymphoid organ, then they will go to the secondary lymphoid organs. Okay everyone? Secondary lymphoid organs. But I will tell you what is the use of B and T lymphocyte. First, step by step only we can go ahead. Step by step. So first you understand this thing, then we will come to the other thing. Right? So secondary lymphoid organ. What is the function of secondary lymphoid organ and what are the examples of secondary lymphoid organs? Beta, these B and T lymphocytes are formed and matured. Now these B and T lymphocytes are going to the secondary lymphoid organs and here they will interact with the pathogen. Here only they will kill the pathogen because pathogens are coming mainly into the secondary lymphoid organs. Okay, so here this is the site of interaction. The secondary lymphoid organs are the site of interaction of B and T lymphocytes with pathogens. Okay, so B and T lymphocytes are interacting with the pathogens where? B and T lymphocytes are interacting with the pathogen where? They are interacting in the secondary lymphoid organs. Alright, so let me give you the examples of secondary lymphoid organs. Let me give you the examples of secondary lymphoid organs, beta. Secondary lymphoid organs. So have you heard of lymph nodes? Lymph nodes. When I will teach you lymph, na beta, lymphatic system. We have two fluids in our body. One is the main circulating fluid, that is blood. From the blood only another fluid is formed, that is lymph. So we have a lymphatic system in which lymph is flowing. In that lymphatic system only we have specialized nodes which are known as lymph nodes. Okay, so lymph nodes. Lymph nodes are the secondary lymphoid organs. Then spleen is a secondary lymphoid organ. Spleen, you know about spleen. Spleen is the graveyard of RBC, right? Then Peyer's patches. Peyer's patches are present in the intestine, beta. They are present in intestine. Peyer's patches. Then you must have heard of appendix. It is also an secondary lymphoid organ. In your pharynx and your your oral cavity and pharynx, you have tonsils. Yes or no? Tonsils. Tonsils are also secondary lymphoid organs. Tonsils. And you have three major tracts. One is your digestive tract, one is your respiratory tract, and one is your urinogenital tract. In these three tracts, na beta, there is a lining of mucus lining. That mucus lining is also a lymphoid organ that we call as malt. Mucosa associated lymphoid tissue. So what is this mucosa, beta? What is this mucosa? Mucosa is the innermost lining of major tracts. What are these major tracts? One is your gastrointestinal tract, one is your respiratory tract, and another is your urinogenital tract. These are the three tracts of the body. Urinogenital means urinary tract also, reproductive tract also. Okay, urinary tract also and reproductive tract also. So in these major tracts there is mucosa, and this mucosa is having the mucus. That mucus traps the pathogens. Okay, the mucus traps the pathogens, beta. And there only the B lymphocyte, the T lymphocyte, they are going and interacting with the pathogen and killing the pathogen. Okay, and killing the pathogen. So now you understood the examples of primary lymphoid organs and the examples of secondary lymphoid organs, right? Based upon the examples only, they can ask you questions that which of the following is not a primary lymphoid organ, or which of the following is a secondary lymphoid organ or primary lymphoid organ, right? So you should know the examples. Bone marrow, thymus, primary lymphoid organ. Others all are here secondary lymphoid organs, right? And here malt, very important line of NCERT. Very important line of NCERT. Malt is forming the 50% of the total lymphoid tissue. 50% of the total lymphoid tissue is malt. Okay, 50%. Malt is forming 50% of total lymphoid tissue. Total lymphoid tissue, because it is present in all the major tracts, na? All the 3 major tracts it is present. So now you at least understood about the immune system. Okay, now you at least understood about the immune system. Immune system is providing you the immunity. So you should know that which system is there, what all are the parts of the immune system which are giving you the immunity. Okay, so now I will be talking about the immunity. Now I will be talking about the immunity. So now let us start with the immunity which is present by birth. Immunity is of 2 types, beta. Immunity is of 2 types. We will classify immunity on various bases. First of all, we are classifying the immunity into 2 types on the basis of whether it is present by birth or whether it is present after birth. So obviously we have a immunity by birth also, and we develop a immunity after birth also. Okay, so we have 2 types of immunity. One immunity is the beta innate immunity. Innate immunity. Innate immunity. Innate immunity. Innate immunity is present by birth, and the another type of immunity is beta acquired immunity. Acquired immunity. Acquired immunity. Okay, so innate immunity is present by birth. Innate immunity is present by birth, whereas acquired immunity is developed or acquired after birth. It is developed after birth. After birth. How it develops? How it develops? When you come in contact with the pathogens, then it develops. Okay, then it develops. So now, if somebody will ask you which type of immunity is non-specific, then innate immunity. Innate immunity is a non-specific type of defense, beta. Non-specific type of defense. Non-specific means for every pathogen, the response will be same. For every pathogen, the response will be same. Like for example, I am teaching all of you same. But some people are understanding, some are not understanding. Some people will get good marks, some people will not get good marks. Right? But I am same for all. Just like that. If the response is same for every pathogen, whether it is a bacteria, whether it is a virus, whether it is a fungi, any pathogen will come, same response will come. Same response. Same response means not specific. If I will treat some of the students specifically, I will tell them something extra points or something, that means I am getting specific for them. And for other students, I am not telling those things, right? Are you understanding the meaning of specific and non-specific, beta? So, acquired immunity is specific. It will treat every pathogen specifically. It will treat every pathogen, differently. So, acquired immunity is specific. It has 4 properties, important properties. 4 important properties of acquired immunity. One of them is specificity. Specificity. That means it is specific for every pathogen. It is specific for particular pathogen. Specific for particular pathogen. Particular pathogen. Right, everyone? Specificity. But here it is non-specific type of defense. Innate immunity consists of some barriers, beta. Consists of barriers. Barrier. I hope you understand the meaning of barrier. Stop. It is stopping the pathogen to enter into the body. It is stopping the pathogen to enter into the body. What is the first thing we have which stops the entry of the pathogen? Tell me in the comments. Tell me in the chat section, What is that first thing we have which stops the entry of the pathogen in the body? Yes, everyone. What do we have? The covering of the body. What is it called? Sai, Raj, Surya. Very good. Skin, skin, skin. Deepti. Yes. Skin, beta. Yes, the Viber, Kritika. Everyone is telling skin. Yes, it is the barrier. It is the barrier. Skin is the barrier. Skin is protective in function. Skin is epithelial tissue. Skin is compound epithelial tissue. Skin is not simple epithelial tissue. Skin is compound epithelial tissue. Compound epithelial tissue only provides the protection. So this is the first barrier. There are 4 types of barriers we will study. The first type of barrier is beta physical barrier. Physical barrier. Physical barrier. The example of physical barrier is your skin. Skin is the physical barrier. Just like that. The next physical barrier is the mucus lining. Mucus lining the digestive tract, mucus lining the respiratory tract, mucus lining the urinogenital tract. Okay. So the first barrier, physical barrier is skin, and another example is mucus lining. Mucus lining. Mucus lining of your gastrointestinal tract, respiratory tract and urinogenital tract. All these tract. Mucus lining. So we have mucus lining in all the major tracts of the body, and we have skin. We have skin covering our whole body. This is the physical barrier, beta. Okay, this is the physical barrier. Next barrier is physiological barrier. Physiological barrier. Physiological barrier means the secretions. For example, we are eating food. We are having food, and in that food there is pathogen. So that food is going into the buccal cavity, right? Saliva is coming into the buccal cavity. Saliva. In the saliva, lysozyme is there. Do you know lysozyme is antibacterial, right? Then it is going into your stomach, beta. In the stomach, you have gastric juice, and that gastric juice has HCl, acid. You know acid. Do you know acid, how much dangerous it is? So acid will, acid is also antibacterial. It will kill all the bacteria. So all these secretions, when we cry, tears, then also tears are coming. But when anything goes into the eye, then also tears are coming, na? So that along with the tear, the pathogen, the dust, that also comes out. Our eyes will be cleaned after crying. Don't cry, although don't cry. But yes, the water which is coming from the eyes, it is cleaning the eyes, okay? It is also having the lysozyme, antibacterial enzyme. So all these secretions of the body, they also protect us, and they are forming the physiological barrier. All these things in us, in our body, are present by birth. We all are covered by birth only by the skin. We all have all these secretions. They have started by from the time of birth, right? Samrudh beta, urinogenital tract is the tract from where the urine is passed, okay? The urinary tract, and genital tract means the reproductive tract, okay? So we collectively call it urinogenital tract, okay? In males it is same, in females it is different. In males, urinogenital tract is same. For urine and for sperm it is the same tract, but in females it is different. Okay, now everyone come to the topic. Physiological. Physiological means you can write here saliva in buccal cavity. You can write HCl in stomach. You can write tear in eyes, tears, saliva, HCl, tears. Then what else? In your ear also there is a ear wax, sebum. That is also a secretion, right? Sebum, right? Sebum, ear wax, ear wax. So when any pathogen is going into your ear, then also it is trapped by this ear wax or sebum, right? So all these are the physiological barriers. Then coming to the cellular barrier, beta. Cellular barrier. Yes, some cells are there. You know these cells. We have already talked about these cells, beta. Cellular barrier. Whenever anything, any pathogen enters into your body, when any pathogen enters into your body, then it first goes into the blood. Yes or no? Blood is the main fluid, na? In the blood, you have the protective cells, the WBCs. The maximum amount of WBCs are neutrophils. Yes or no? The majority of the WBCs are neutrophils. They will do the phagocytosis. Yes or no? So these are the cellular barrier, beta. Cellular barrier. In your blood they are present. Cellular barrier. So cellular barrier consists of the cells. They can be phagocytic cells. They can be phagocytic cells. There can be some non-phagocytic cells. There can be some non-phagocytic cells. I will give you the example of phagocytic cells first. Phagocytic cells are your neutrophils, beta. Neutrophils can do phagocytosis. Then you also have monocytes. When these monocytes goes into the tissue, they become macrophages. They become macrophages in tissues. In tissues, when you read NCERT, you will come across this. Monocyte is present in the blood. But when this monocyte enters into the tissue, it becomes macrophage. So neutrophil, monocyte, macrophage, they are phagocytosis. They are doing what? Phagocytosis. So they are phagocytic cells. One more cell is written in NCERT, that is natural killer cell. Natural killer cell. It is a killer cell, beta. It will kill the pathogen. It will kill the cell in which pathogen is there. Okay, natural killer cell. It is a non-phagocytic cell. It is a type of lymphocyte. It is also a type of lymphocyte. It is a type of lymphocyte. Very, very important. So these are the cellular barriers. All the cells in the blood, they are the cellular barriers. Now, the next thing is the cytokine barrier. Cytokine. Cyto means cell, kine means it is secreting something. Cytokine barrier. So for cytokine barrier, you should remember one example, that is interferon. Cytokine is secreted by virus infected cells, beta. Virus infected cells. Virus infected cells are secreting the cytokine. You know, they are very, very, selfless. They are not selfish. They are not selfish. If let us suppose there is a cell and virus has come and infected that cell, now this cell has to die because virus has infected, but this cell will protect the other cells from the viral infection. How it will protect the other cells from the viral infection? It will secrete some secretion, and that secretion is only called as cytokine. Okay, so there can be so many different types of cytokines. One example is given in NCERT, that is interferon. Interferon. So the example is interferon. You should remember this example, interferon. Example, interferon. Interferons. IFNs. Interferons. So virus infected cells are secreting something that is known as cytokine. Example is interferons. They are secreting interferons, beta, and these interferons are going to the other cells and telling them that, hey, all of you, virus has entered into the body, so you should stay safe. Okay, so it is alerting the other cells, and the other cells, they have their own mechanism so that they will protect themselves from the viral infection. Isn't it interesting? Yes, sacrifice. Yes, Ramya Rao. Sacrifice. All right. All right. So, you know, our body is very amazing, and few things in our body are so amazing that they teach us life lessons also. Hai na? So what they are teaching, the virus infected cells, they are teaching us not to be selfish. Okay, they are teaching us that we should be humble, kind to others. We should care for others. All right, we should care for others. All right, such good cells, Malika Arjun. Yes, beta. All right, now you understood the barriers. So all these barriers are present in our body by birth, and they are forming our immune system. Which immunity they are providing? They are providing innate immunity. If you all have understood, please give a thumbs up. Innate immunity. Have you understood innate immunity? Quickly, I will tell you the acquired immunity quickly. Okay. So coming to the acquired immunity, I have told you 4 important properties. First is specificity. Second is diversity. Diversity. I will tell you what is the meaning of diversity. Third is discrimination. And the very important 4th property is beta property of memory. Okay, so all right everyone, all right, I'm glad you all have understood. I'm glad. Now focus on the acquired immunity. Specificity means specific. You understood that. Now diversity means lagging. Is there any lagging? Thank you, Ganesh beta. Thank you. Okay, Sushant, maybe it is the problem in your network beta. Okay, in others everything is fine. All right, so now everyone, diversity means it can recognize variety of pathogens. Diversity means it is not like it is recognizing only bacteria, only virus, only fungi. No, it can recognize variety. Diversity means variety. For example, you like variety of foods, है ना? You like variety of snacks, so you like diversity of snacks. Okay, you like diversity of animals, right? We have studied diversity of animals in the animal kingdom, just like that. Okay, thank you beta, thank you Sai Subramanyam. So diversity means can recognize variety of pathogens. Can recognize variety of pathogens. Okay, coming to the discrimination. Yes, discrimination is there. This acquired immunity can distinguish between what is mine and what is not mine. Okay, what are self cells and what are non-self cells. If there are some cells which are coming into the body, it will recognize that these does not belong to my body, right? And what are self cells? It will protect the self cells and it will kill the non-self cells. Okay, so discrimination means can distinguish or differentiate between self and non-self. Self and non-self beta. Self and non-self. Thank you beta. Thank you Sheikh Nazia. All right. Thank you Bhashwant. All right. Memory. I am talking about the very important property, the most important property, that is memory. Okay, memory. For example, you are meeting a person for the first time. Okay, let us suppose that you are meeting any person for the first time. So what will be your response? For example, when you will come here, you will make new friends, you will come here. So then when you are meeting a person for the first time, your response will be very low. You will start talking very, slowly. You will be interacting much. You won't be interacting much in the first meeting. You will be saying, okay, what is your name? How are you? From where you came? Like that. But when you come to know that person and you are meeting that person the second time or third time, you will say, hey, hi, how are you? You will say like that. So that means we are giving 2 types of response. First response is the primary response. Primary response is a low intensity response. Primary response is the response we are giving when we are meeting anybody for the first time. But when we know anybody, when we know and when we meet the known friends, when we meet the known people, our response is of high intensity. hi, how are you, है ना, like that. So our response is of high intensity, that is secondary response beta. So memory means our immune system can have the memory of first encounter. Memory means can remember first encounter. So what is the first encounter? What is the primary response? Two types of response due to the memory. Two types of response beta. This will be the last thing which I am going to tell you. Two types of response. One is primary response. Primary response is given when the pathogen is coming into your body for the first time. Your body is not prepared and the response will be of low intensity. So primary response is when pathogen comes for the first time. When pathogen comes for the first time. So this response will be of low intensity response. But when the same pathogen comes for the second time, right? When the same pathogen comes again, maybe it is the second time or third time or fourth time. Whenever this same pathogen is coming again into your body will give high intensity response. So secondary response is given when the same pathogen comes again, right? Again, and it is of high intensity response. So it has a fancy name beta. It has another name, fancy name, that is known as anamnestic response. Another name, important term given in NCERT. Anamnestic response is the secondary response. Alright. So this you have to remember. Why high intensity response came? Due to memory. Why this high intensity response came? Due to the memory of first encounter. So this is the important property, memory, right? I hope you have understood the meaning of primary response and secondary response. This type of response you will not see in innate immunity, you will see in acquired immunity. I hope you have understood the concept of immunity. Further, many types of immunities are there. Once you come here, we will understand about the immunity more. And there is a lot to teach, there is a lot to understand. Okay, so stay tuned. This is the end of today's class beta. I know that I have taken a little bit more time today, but I know that you will adjust, है ना? So now you enjoy the session is over. You can like the session. Always don't go without liking the session. All right, bye bye, take care beta. God bless you all and see you tomorrow. Bye bye.